Hello everyone, my name is Snehlata and I'm a PhD student and a PMRF fellow in the Department of Biological Sciences and Bioengineering, IIT Kanpur. Today we will continue with the next lecture of the chapter Biomolecules. So till now we have studied about one major biomolecule, that is protein, and its monomer, that is amino acids. So in last lecture we studied about the protein structure of proteins, right? So before moving to the next lecture or the next topic today, we will quickly solve two questions related to protein. So the first question is, which of the following statement is true about the primary structure of proteins? So what we saw in the earlier class that protein has four structures: primary, secondary, tertiary, and quaternary. And the most native structure is primary structure. So what options do we have? The first option is that it is a helical structure of a protein. So what we saw that the helical structure of a protein belongs to secondary structure, right? So it is not a primary structure. The second option is subunit structure of a protein. So we saw that the subunit structure of a protein is quaternary structure or 4-digit structure in which we have different subunits. The third option is three-dimensional structure of a protein. The three-dimensional structure of a protein belongs to tertiary structure, right? So 3-degree structure. And the final option is the sequence of amino acids joined by a peptide bond, which is a correct answer. So if you remember, the primary structure is just a chain of amino acid that are joined together by the peptide bond. So it is a linear chain. Now, the second question is, which of the following statement is true about proteins? So you can also consider this question as a role of protein. You can also think it as a role of proteins. So proteins are most abundant biomolecules in our cell and they have function everywhere. So let's look at the option. So the first option is proteins are made up of amino acids, which is correct. Second is proteins are essential for developing skin, teeth, and bones. That is also true because proteins play a very important role in all the structural support that our body have. The third is protein is the only nutrient that can build, repair, and maintain body tissue. This is also a correct answer. Along with others, protein is also the nutrient that can perform all the functions. Okay, so it is the only nutrient that can perform all the functions, like it can build, repair, and maintain the body tissues. So the correct answer will be all of the above. So if someone asks you about the role of the protein, you can also write these options. Now, after completing protein, now let's move to the next topic, that is enzyme, which is also related to protein. So what are enzymes? So enzymes are made up of proteins. Okay, enzymes are made up of proteins, or you can also say enzymes are proteins. But is all enzyme protein? The answer is no, because we have although most of the enzymes are protein, we have two exceptions. Okay, and the first exception is we have ribosome. So we have ribosome. This is an enzyme, and this enzyme is present. This enzyme is present in ribosome. So remember, ribosome is an organelle that is present in both prokaryotes and eukaryote, and it is involved in protein synthesis, right? And this ribosome is present in ribosome, and this ribosome is of RNA nature. So it is not a protein, it is a RNA, but it acts as an enzyme. Now, the second exception we have is ribonuclease P. The second exception is ribonuclease P. And both of these enzymes are what? Their nature is, although most of the enzymes are protein, but the nature of these two enzymes is RNA or ribose nucleic acid. Okay. So these are two exceptions of enzyme. Apart from this, all the enzymes that we know are made up of proteins. Okay. Now, also this ribosome, it is part of which RNA? It is a ribosomal RNA. So we will study about types of RNA, but for now, remember that it is ribosomal RNA, okay? Because it is present in the ribosome. Now, If we talk about enzymes, so in what pool these enzymes will be present? Will it be in acid soluble pool or acid insoluble pool? So if you remember, because it is made up of proteins and the proteins belong to acid insoluble pool, the enzymes will also belong to acid insoluble pool. So they will be present in acid insoluble pool or retintate Okay. Nextly, we know because it is made up of protein, we know protein has four types of structures, like primary, secondary, tertiary, and quaternary. So, what is the most occurring structure of an enzyme? So, the most occurring structure, most common structure, most common structure of enzyme is tertiary. Okay, is tertiary structure. Okay, so most common occurring structure is tertiary structure. Then enzymes also exist in primary, secondary, but the most common is tertiary structure. And if you remember, tertiary structure is what? We have a linear chain of primary structure that has this N terminus and C terminus, right? This structure folds to form something like this, alpha helix or beta sheet. And this is what? This is your secondary structure. And when this structure folds upon itself, it folds upon itself to form a structure something like this. This is your tertiary structure. So the enzyme mostly exists in tertiary structure. Now, what is the nature of this enzyme? So this enzyme is globular in nature. So we had also studied about types of enzyme based on their types of proteins based on their shape, right? And we saw that there are two types of proteins, globular and elongated or fibrous. So the enzyme is of globular nature. And if it is globular nature, it means it is soluble. It is soluble in water. It is soluble in water. So enzymes are soluble in water. Now, what is the source of enzymes, or from where can we get enzymes? So the original source of enzymes is living cells. Okay. Enzymes are synthesized and are found in living cells. But does it mean that it can only work in the living cell? The answer is no. You can always isolate an enzyme and you can use it for your own purpose outside the cell. So the enzymes, if you provide optimum condition, enzymes can also work. Although they are produced inside the cell, but enzymes can also work outside the cell. Okay, outside the cell. But under what condition? Under optimum, or the optimum means the desired condition of this enzyme. It can also work outside. Okay, so this is a general overview about the enzymes. Now let's talk about the nomenclature of enzyme. The nomenclature, as you all must be knowing, it means naming. So naming of the enzymes. So the question is, why do we need to name enzymes? The answer is, if you look at your body, there are large number of chemical reactions going on in the body, and these reactions are catalyzed by the enzymes. Okay, so if you start naming each of these enzymes, it will be very difficult to remember. So scientists have come with certain nomenclature or certain way of naming these enzymes. So before talking about that, first let's see that who discovered the enzyme. So enzyme was first time discovered by a scientist named Edward Buchner. So enzyme was discovered by Edward Buchner. Okay, and how did he discover the enzyme? So he did not specifically targeted to discover enzyme, but it was discovered in serendipity. It was discovered in serendipity. One sec. So it was discovered, serendipity means by chance. Okay, so it was discovered by chance, or you can say serendipity. Serendipity or by chance. Okay, Now, let's talk about the nomenclature. So, like I said, why do we need nomenclature? Because there are a large number of enzymes, and it's very difficult to remember the name of each of these enzymes. Large, just like organic chemistry. In organic chemistry, we have large number of compounds, and hence we came up with a certain way of naming those compounds, right? The IUPAC nomenclature, right? Similarly, the enzymes also have a certain way of naming, and this is known as IUB nomenclature that we'll talk shortly. So, before IUB nomenclature, people also tried naming enzymes by different way, and one of such ways to use suffix, to use a suffix. Suffix means the letter that is added at the end of the name. So they try to use a suffix ase. Okay. So they try to use a suffix ase, such as example, if you talk about amylase, amylase is an enzyme. Okay. So what people said that let's add a suffix ase after any name. But this was not feasible or widely acceptable. Then people's second approach that they used was to name these enzymes after their source, because we know that enzymes are produced from a living system. So the second way was source. So from where this enzyme is isolated. For example, if it is isolated from, say, papaya, so the enzyme name will be papain, because it is isolated from papaya. But this is also not feasible, because in English we call it papaya, in some other language we will call it some other name. So this nomenclature was also not feasible. Then third nomenclature that is most widely accepted is IUB nomenclature. So what is IUB nomenclature? So IUB stands for International Union of Biochemistry. So it stands for International Union of Biochemistry. And they give IUB nomenclature to enzyme, and this nomenclature is known as EC number. So each enzyme was given a EC number. Now, what is EC number? So EC stands for enzyme commission number. Okay. So EC stands for enzyme commission number. And this nomenclature by IUB is most widely accepted. It is most widely accepted. Okay. So according to IUB nomenclature, each enzyme is given a EC number, and the EC stands for enzyme commission number. Now, what do we have in this EC number? So this EC number has each enzyme was given 4 numbers, was given or represented by 4 numbers. Okay, 4 numbers. For example, suppose our enzyme is named like 4.3.1.2. So in the similar fashion, all the enzymes are named according to this IUB nomenclature. Now, what does this number denote? So the first number, it denotes the class of that enzyme. Okay, so it denotes the class of the enzyme. It denotes class of enzyme. So based on the function, the enzymes are divided into different classes, right? So it is one second. So the first number represents the class of enzyme. Now the second number. So the first number was 4. So 4 represents the class of enzyme. The second number represents subclass of enzyme. It represents subclass of enzyme. Okay. And then the third number. What does third number represent? The third number represents subdivision of the enzyme. Third represents a subdivision of the enzyme, and the fourth represents the reaction catalyzed by the enzyme. Okay, so fourth, it represents the reaction, the specific reaction catalyzed by that enzyme. Catalyzed by the enzyme. Okay, so these are the four numbers that represent different category of that enzyme. Now, what is important to us is the class of enzyme. So for now, it is what is important is the classes of enzyme. So according to IUV, the enzymes are divided into 6 classes. So enzymes are divided into 6 classes. Now let's see one by one what are these classes. And the most important point to remember is that you have to remember these classes in the same order that I'm going to write. Because what we saw here, that the first number here is representing the class, right? So the 4 here is representing a class. So this enzyme will belong to a class that is on the 4th number in the order. So let me first write the classes of enzyme, then I'll explain it to you by an example. So the first class of enzyme is oxidoreductase. One second. So the first class, or you can say 1. So it is oxidoreductase. First class of enzyme belong to oxidoreductase. Now, what does oxidoreductase do? So it catalyzes oxidation reduction reaction. So we know what is oxidation reduction, right? So it catalyzes oxidation reduction reaction. So what is oxidation? In general terms, oxidation is gain of oxygen or loss of electron. Reduction is gain of hydrogen or gain of electron, right? So oxidoreductase, what it do? It catalyzes the oxidation reduction reaction. Let's see an example. What I'm trying to say. So we know that oxidation is what? Gain of electron. Gain of oxygen atom or lose of electron, right? So suppose you have a substrate that is I'm denoting by S, and it has an oxygen attached to it. Then you have a different substrate that I'm representing by S dash. Now the function of this oxidoreductase is, so if the reaction is taking place in the presence of oxidoreductase, what it will do? It will perform this oxidation reduction reaction, right? So one of the substrate will get oxidized and the second will get reduced. Here, which of the substrate is oxidized? If you see, oxygen is attached to it. So here it is oxidation, or this S is oxidized. And this is in the reduced form. Okay, it is in the reduced form. But when the oxidoreductase come into picture, what it do? It performs the oxidation reduction reaction, and it will do? It will remove this oxygen from here, and it will bind this oxygen to this S dash. So now this is oxidized, and this S is reduced, and this reaction is catalyzed by oxidoreductase. Understood? So the oxidoreductase, what it do? It catalyzes oxidation reduction reaction. And what are the examples of oxidoreductase? The examples are catalase. So catalase is an enzyme, or peroxidase. So these are certain enzymes that belongs to first class or oxidoreductase. Okay. Now, which is the second class? So class 2. What is class 2? Class 2 is transferases. Class 2 is transferases. So this is first class. Class 2 is transferases. So as the name suggests, transferases is transfer something. Okay. So what it does is it transfer group, a functional group, from one substrate to another, from one substrate to other. Okay, so it transfers group, or you can say functional group, transfers functional group from one substrate to other. Let's look at by an example. So again, suppose this is a substrate and it is attached to a functional group X, and then there is another substrate S dash. Now, in the presence of this transferase, if we take in the presence of this transferase, what it will do, it will remove this group from here and then attach it here. Okay, so what we will have? This thing. So it has transferred the group. And the example of transferases are kinases. The kinases, what does kinase do? It transfer the phosphate group. Okay. Another example we have is trans-aldolase. Trans-aldolases. So these are the examples of class 2 or transferases class of enzymes. Now the third class is hydrolases. Okay. So class third we have is hydrolases. Now what are hydrolases? It has hydro, means water in it. So what does hydrolases do? So they break bonds. Okay. Hydrolases, they break bonds, but in the presence of water. So they uses water, okay, to break a bond. So they break bonds in the presence of water. In the presence of water, or by utilizing water molecule. Okay. So this is a function of hydrolases. Let's look at it as by an example. So suppose this is a substrate. It is bind to this group A. Now, this hydrolases, what it will do? The hydrolase, what it will do? It will use water molecule. Okay, it will use water molecule to break this bond. And the resulting product will be this, because this water has what? H plus and OH ion, right? So it will use this water to break the bond, and the resulting product will have OH group attached to it, and then the remaining H will be attached to H. Okay. So by this way, this bond between S and A is broken by hydrolases. And if we look at the examples, what example we have? We have amylase. We have amylases. We have all digestive enzymes. So all digestive enzymes such as trypsin, chymotrypsin. So all the digestive enzymes, they belong to this class third or hydrolases group. Now the next class is class fourth. The next class is lyase. Okay, the next class is lyase. Now, what does lyase do? So lyase, again, it also break bonds. Okay, but the difference is it break bonds, but the difference is that it does so without using water molecule. So it break bonds without using water molecule. Okay, without using water molecules, right? So if we have an example, and also along with breaking the bonds, it also introduces double bond. So it breaks a single bond and introduces double bond. Okay. So suppose this is S that is attached to X. You have S test that is attached to Y. So if the reaction, so this is bond. Okay. So if lyase is present, what it will do? It will break this X and Y bond from S and S test, and it will result in the product that will be something like this without using water. So it has broken this already existing bond between S, X and S test Y and introduced this double bond between S and S test. Okay. And the examples of this class are aldolase. Aldolase. So most of these enzymes belong to the glycolysis pathway that we will see later. But for now, so they belong to aldolases or carbonic anhydrase. Carbonic anhydrase. So these are class fourth enzyme or the lyase group enzyme. Now the next class is class 5, and it is isomerase. Next class is isomerase. So what do we have in isomerase? So it is derived from that word isomers. Okay. And what are isomers? having same chemical formula but different structural arrangement, right? So they isomers have different structural arrangement, and this is what isomerase do. Isomerase, what it does, it changes the structural arrangement of the molecule, changes structural, it changes structural arrangement of molecule. Okay, and if the structural arrangement is changed but this chemical formula is same, it will lead to the formation of isomers, right? So leads to the formation of isomers. So isomerase are responsible for the isomers formation, right? So it leads to the formation of isomers. So, for example, you have glucose 6-phosphate, where glucose 6-phosphate. So this glucose 6-phosphate, in the presence of isomerase, it gets converted into fructose 6-phosphate. So glucose and phosphate. Okay, so these are isomers. In the presence of isomerase, this glucose 6-phosphate is converted into fructose 6-phosphate. And the example of isomerases are phosphohexoisomerase. One of the example is phosphohexoisomerase. Okay, and now lastly, class 6th. So what is class 6th enzyme? Class 6 enzyme belongs to ligases. So there is difference between liases and ligases. Okay, so these are ligases. These are ligases. Okay, so what does ligase do? It simply forms new bond. It simply forms new bonds. So if there are two substrate molecules, they are in isolation, this ligase forms new bonds. For example, what kind of bonds it forms? CC bond or CN bond, CO. So these kind of bonds this ligase forms. And the example is DNA ligases. So DNA ligases play a very essential role in DNA replication. We will see it in upcoming lectures, but for now, just remember that DNA ligases belongs to this group of ligase that play essential role in DNA replication. So with this, we have completed the classes of enzymes. Now let's go back to our previous example that I was saying. So the example I had is, suppose an enzyme is given a name something like this, 4.3.1.2. And if someone asks you that of what class this enzymes belong, so because it has four number, so it will belong to class 4. And what is class 4? The class 4 is lyase, right? So because of this reason, you have to remember this enzyme class in its original order. And for this, you can also remember it like OTH, LIL. So by this, you can remember OTH, LIL. Okay. So oxidoreductase, transferase, hydrolases, lyases, isomerase, and ligases. So if someone asks you what is class 4, you can simply answer lyase, right? So now, since we have seen the nomenclature, now let's move to the structure of enzyme. So the structure of enzyme, it is similar to that of a protein, and mostly it occurs in the tertiary structure. So what we can say, that first DNA will encode the primary structure of enzyme, right? So DNA, if someone asks who has this information about the primary structure of enzyme, DNA has this information. So DNA encodes primary structure of enzyme. Now this structure will fold to form secondary structure, and then this secondary structure on itself fold to form a structure something like this, right? So it will have this globular structure. So it will have this globular structure something like this that is represented here. Now what we can see in this structure is that this structure is forming these pockets. So you can see there are these pockets again there is this gap or pocket, right, on which anything can come and bind. So this tertiary structure, what it has pockets. It has pockets, or you can also say cleft. Okay, so this tertiary structure has pockets. So this is your tertiary structure or the structure of enzyme, and it has what? Pockets or cleft, right? On which anything can bind. Now, what is the function of enzyme? The enzyme catalyzes the reactions, right? And for the reaction, we need two things. What do we need? We need a substrate and this enzyme itself. So what we need? We need a substrate. So suppose at this pocket, if a substrate bind, then this pocket is known as active site. Okay. So for enzyme, what do we mean by active site? So what is active site? Active site, it is a site or pocket of enzyme. It is a site or pocket of enzyme where substrate binds. Where substrate binds and reaction takes place. Okay, and reaction takes place, right? So active site is a site or pocket of enzyme where substrate bind. So although enzyme has multiple pockets, but there will be a specific pocket on which a particular substrate bind, and that site is known as active site, where the substrate binds and the reaction takes place. Okay. Now, next, let's talk about the function of enzyme. So we have seen the structure of enzyme. The enzyme is nothing but it's a tertiary structure which has pockets to which a substrate binds. And now let's talk about the function of enzyme. So what does enzyme do? So like I was saying, the enzyme, it catalyzes the reaction in the cell. It catalyzes the chemical or metabolic reactions. Chemical or you can say metabolic reactions in the cell, right? So it catalyzes chemical or metabolic reaction in the cells. All the chemical and metabolic reactions are catalyzed by enzymes. And what do I mean by this catalysis of chemical or metabolic reaction? So let's see it by an example. So suppose you have glucose. And then this glucose is getting converted to pyruvate. It is getting converted to pyruvate. So here the substrate is glucose. So glucose is what? It is substrate. And what we want from this glucose? We want pyruvate. So this pyruvate is our final product or the end product. Now, in order to convert this glucose to pyruvate, there will be a certain metabolic reactions. And this metabolic reaction will be catalyzed by an enzyme. So the enzyme will catalyze this metabolic reaction, and in which this is a substrate that will bind to this enzyme, and then this enzyme will cause certain changes in this substrate that leads to the production of this product, that is pyruvate. Okay, so the substrate we also represent it by S, and the product we represent it by P. Now, the important feature of the enzyme is it increases the rate of reaction. Okay, it increases the rate of reaction. Now, what do I mean by increasing the rate of reaction? So the rate of reaction means you can also say speed or velocity. So speed or velocity of the reaction. Velocity if it has direction. Okay, so it increases the rate of reaction or speed or velocity of reaction. Okay, now what do I mean by rate? The question is, what do I mean by rate? So the rate means that how many product molecules are being formed per minute or per unit time by an enzyme. This is known as rate. Okay, so rate means number of product. It means number of product molecules formed per unit time. Formed per unit time by a enzyme molecule. By a enzyme molecule. Okay. So this is rate. Rate means, or you can also represent it by like delta P by delta T. So P is product, T is time. So the number of product molecules that is formed per unit time by enzyme molecule is known as rate. And the function of enzyme is to increase this rate of reaction. Okay. So suppose initially without enzyme, example, without enzyme, suppose without enzyme, you have say 100 substrate molecules that are converted into 100 product molecules in say 1 hour. But if there is an enzyme, so the function of enzyme is it will increase this rate of reaction. Okay. So the function of enzyme is it will increase this rate of reaction. So now in this 1 hour, you will have, it can utilize say 10,000 substrate molecules in this 1 hour, and it can convert this 10,000 substrate molecules into 10,000 product molecules. So it has increased the reaction rate approximately 100 times, right? So this is what I mean when I say that enzyme increase the rate of reaction. Now, the second thing, what does enzyme do, or how does it increase the rate of reaction? So it causes certain changes in the substrate, right? So enzyme cause changes. It causes changes in substrate. And what are these changes? So the first change is physical change. Okay. So the first change is physical change. So what do I mean by physical change? Physical change means it changes the shape of the substrate. So changes shape. So the change means shape, or it can also change the phase of the substrate. So shape, suppose it has initially this shape. Now this enzyme has changed its shape to something like this. Okay. Also by phase, what I mean, suppose it is initially in the solid phase, the enzyme change it into liquid phase. Okay. So this is what I mean by physical change. Secondly, the substrate also the enzyme also causes chemical changes. So it also causes chemical changes. Chemical changes mean it break and form new bonds. So it break already existing form and form new bonds. Okay. So the enzymes can cause both physical and chemical changes to the substrate. So this is what is the function of enzyme. It catalyzes the reaction and increases the rate of that reaction. Okay. Now, how does this enzyme perform this function? So how does the enzyme perform this function? So the enzyme, first of all, this enzyme, because we know that enzymes are globular in nature and they are soluble in water, right? So they are globular in nature. So they perform their function in the aqueous solution. Okay, so in order for an enzyme to perform certain function, it needs to be an aqueous solution. So the enzyme performs function in aqueous solution. Okay, in aqueous solution. Okay, so our enzyme needs to be in the aqueous solution to perform the function. Now, if the enzyme is present in the aqueous solution, how would a substrate finds it, and how would a substrate binds to its active site? Okay, so a substrate binds to, how does substrate bind? So substrate binds to enzyme active site, by diffusion. Okay. So what I mean by this is, suppose you have a lot of substrates like this. You have a lot of substrate in the aqueous solution, and then you have this enzyme. Okay. So the substrate will, and this is all aqueous solution. This is everywhere is an aqueous solution. So this substrate will come and bind to this via diffusion. Okay. It will bind to the active site. This is the active site. And here substrate will bind via diffusion. Okay. Now, what happens? So, first of all, the substrate will come and bind to this active site via diffusion. Now, once this substrate has bound to this enzyme, say substrate has bound to this enzyme, right? This is your enzyme, this is your substrate, this is your enzyme that I am representing by E. So, it will form, and then upon binding, there will be certain changes in the active site. Okay. So, there will be change in the shape of active site. There will be change in the shape of active site, and this change will lead to the tightening of the active site. Okay, so now the substrate has been bound tightly, so it will cause change in active site shape, and due to this change, the active site or the enzyme will tightly bound this substrate, and it will lead to the formation of a complex, and this complex is known as ES complex. So what is ES complex? ES complex is nothing but enzyme substrate complex. Okay. Now this enzyme substrate complex will undergo change or catalysis, and this substrate will be converted into product. So this substrate, suppose this was a substrate, now this substrate will be converted into product. Okay. This will be converted into product. So this will be your product now, and this is your E. So this will lead to formation of EP complex, enzyme product complex. And then this enzyme product complex, from this enzyme product complex, the product will be released. So the product will be released, and the enzyme will be again ready for the next reaction. So this will be your E, this will be your P. So this complex will be called as E plus P complex, enzyme and product complex. And this is EP complex, in which enzyme is still, the product is still bound to enzyme. And what is ES complex? This is the complex where substrate is bound to the enzyme. Okay. So this is how the enzyme functions. So what we saw, that in the enzyme, the first step is that substrate binds to the enzyme active site via diffusion. After this, what happens? After this, it induces, after binding, it induces change in the shape of active site, that leads to the tightening of substrate by enzyme, right? So enzyme tightly binds to the substrate, and it leads to the formation of ES complex. Now, after this, what happens? After this, what we saw, that substrate to product formation takes place. Substrate to product formation takes place, and then product is released. Right? So these are the 4 major steps by which the enzyme functions. Now, in between these steps, if we want to write a chemical equation, how we can write? So we can write that initially what we had E plus S, right? We have enzyme, we have substrate in the solution. Now this ES will come and bind, this substrate will come and bind to enzyme, and this will lead to formation of ES complex. Now remember, this step is reversible. Okay. So after binding to the substrate enzyme, the substrate can also again get released and can come into this E plus S complex form. Okay. So this ES complex, this is also known as transient state. Okay. So this is temporary or transient state. Okay. Now, this reaction can go either way, either backward or forward. So, but once this, after this, if it is going forward, it will lead to the formation of EP complex. Okay, and this is one directional process. So once this EP complex is formed, it can only go forward, it won't go backward. Okay, so this is an enzyme product complex, and then this enzyme product complex will break and this product will release, and it will lead to formation of E plus P complex. So this is the equation that we can see if we want to represent in the equation form. So this is how it can be represented. And in this, the one step that is reversible is this E plus S2 ES complex formation. Okay. Now, so this is how this enzyme works. Now we have talked about the enzyme function. How does it function? Now let's talk about the features of enzyme. So what are the features of enzyme? So the first feature of the enzyme is about the nature of the enzyme. The first feature is nature of the enzyme. So we know what is the nature of enzyme. It is mostly proteinaceous. We know the exception. So it is mostly proteinaceous, that is made up of protein. Okay. And the second thing is it is globular. It is globular in nature. Third and important thing, as all catalysts, because enzyme is what? It is catalyst. So it increases the rate of reaction, but it does not get used up in the reaction. Okay. So it is not getting consumed in the reaction. The concentration of enzyme before and after the reaction will be same. Okay. So this is an important feature. It will not come under nature, but this is an important feature of enzyme, that enzyme does not get consumed in the reaction process. Okay. So it catalyzes the reaction, but it does not get consumed in the reaction. Okay. Now, so the nature, it is proteinaceous and globular. Second thing is about a term known as turnover number. So the turnover number is specific for a specific enzyme. Now, what is turnover number? So the turnover number means that in a minute, how many molecules of substrate are getting consumed or are getting converted into product by a single molecule of enzyme. Okay. So in a minute, like I said in the previous example, that here the substrate comes and bind to the enzyme, then this substrate is getting converted into product. And once this product is released, this enzyme again gets ready for binding with another substrate. So how fast this can be done, which is determined by turnover number. So turnover number is number of substrate. So in a minute, how many substrates are binding to this enzyme and getting converted to the product, that describes the turnover number. So it is number of substrates converted into product per minute. Okay. Per minute by a single molecule of enzyme. By a single molecule of enzyme. Okay. So this is the turnover number, that how many number of substrate molecules are getting converted into product per minute by a single molecule of enzyme. Okay. And the enzymes usually have very high turnover number. Enzymes have very high turnover numbers. Okay. Now, so this is the second feature of enzyme, that is its turnover number. Now, what is the third feature? Third feature is specificity. Okay. So what do we mean by specificity of enzyme? So specificity means, because we know that enzyme is present in our cell, right? But how does this enzyme know what substrate to bind? Can it bind to all the substrate or any of the substrate randomly? The answer is no. So the enzymes specifically bind to a specific substrate. Okay. So specificity means that enzymes are highly specific in the term that they bind to a specific substrate. Okay. So enzymes are specific to a particular substrate. Okay, to a particular substrate. So enzymes cannot bind to any of the substrate, but then specific to a particular substrate. And how does they know that they have to bind to this specific substrate? It is due to their shape. Okay, due to their shape. Now, what does I mean? So what do I mean? I mean that this we saw that the enzyme have this active site, right? And this active site is of certain shape. So this enzyme, suppose this is an enzyme, okay, and this enzyme, what it has? It has active site, right? It has active site. Now you can see that this there is certain shape of this active site. Now this active site of the enzyme will bind to only that substrate that has a shape complementary to this active site. So suppose you have this substrate, you have this substrate, you have this substrate. So this enzyme will not bind to this or this. It will not bind to this or this because it has shape complementary to this. It will bind only to this substrate, and because of this, it has this specificity. Okay. So what we can say? We can say that it is specific because the shape of substrate is complementary. Shape of substrate is complementary to the shape of enzyme active site, to the shape of enzyme active site. Okay. So because of this reason, the enzymes are highly specific because they bind to a specific substrate. So this is the third feature of enzyme. Now, what is the fourth feature of enzyme? The fourth feature is selectivity. So the fourth feature is selectivity. Okay. So let's talk about the selectivity of the enzyme. So what do we mean by selectivity? Selectivity means that a single substrate can be bound to multiple enzymes. Okay, because multiple enzymes can have a shape complementary to the shape of that substrate. So I'm talking about single substrate. Okay, but those enzymes, the product that they will form will be different. Okay, so what I'm trying to say that a specific enzyme, but what I'm trying to say is that a single substrate can form multiple enzymes. Can form multiple products. For example, we have pyruvate, right? We have pyruvate. Now, this pyruvate can form lactic acid. It can form lactic acid. It can also form acetaldehyde. It can also form acetaldehyde, and it can also form acetyl-CoA. It can also form acetyl-CoA. Okay. And now this pyruvate has certain specific shape. So it can bind to an enzyme that has the shape complementary to its active site, right? But the end product that will be formed, the product, these are the products, right? The pyruvate is a substrate, and these are the products. So the products that will be formed can be any of these three, like lactic acid, acetaldehyde, or acetyl-CoA. So does it mean that a single enzyme can form all these products? The answer is no. Okay. So this is what the selectivity of an enzyme means. It means that a specific enzyme, so the selectivity, what I mean by selectivity is specific enzymes, that specific enzymes make one product or one type of product from a substrate. Okay, so it means although this pyruvate can get converted to any of these three products, but a specific enzyme, so if there is say enzyme X that is responsible for forming lactic acid, so when it will bind to pyruvate, it will only form lactic acid, not acetaldehyde or acetyl-CoA. For acetaldehyde, you will have different enzyme. For acetyl-CoA, you will have different enzymes. Okay. So a specific enzyme make one type of product, only one type of product from a substrate. Okay. And this is what is selectivity of an enzyme. Now, the fifth thing about a feature of this enzyme is metabolic pathway. So the fifth thing is metabolic pathway. So what do I mean by this? So what I mean is that the enzymes link the substrates in such a way that it leads to the formation of a metabolic pathway. So series of enzymes link metabolites They link metabolites. What are metabolites? These are the intermediates that are formed in a metabolic pathway. So series of enzymes links metabolites and it leads to the formation of metabolic pathway. And it leads to the formation of metabolic pathway. Metabolic pathways. Okay. So for example, suppose you have this substrate and this is your enzyme. Say this is your enzyme. Now this substrate when binds to enzyme gets converted into say a product like this. Okay. Now this product will act as a substrate for another enzyme that has this active site something like this. And when this enzyme convert this product into say a structure something like this. Okay. Now it will be used as a substrate. This enzyme. So this product will be used as a substrate for another enzyme. So it will be used as a product, a substrate for another enzyme. And this way, a chain of reactions will happen that will lead to the formation of metabolic pathway. So it means that product of one enzyme will act as substrate for another enzyme. Okay, and this will link this complete metabolites and it will lead to the formation of metabolic pathways. Okay, so these are the features of enzymes. Now let's quickly look at the difference between enzymes and inorganic catalysts. So what I say, enzyme is an catalyst, right? And we also have seen in the chemistry that there are certain inorganic catalysts also. And the function of catalyst is what? That it increases the rate of reaction, right? So the enzymes, these are called as biocatalysts. Enzymes are called as biocatalysts. Why? Because they are isolated from the biological source and they catalyze the biological reaction. So they are called biocatalysts because they catalyze biological reactions, right? They catalyze biological reactions. And because of this reason, they are called as biocatalysts. So now let's quickly see what is the difference between biocatalysts and inorganic catalysts. So the first difference we saw. The first difference is that it catalyzes the biological reaction, while the inorganic catalyst, they catalyzes the other reaction that is outside the cell. Although this biocatalyst can also catalyze the reaction outside the cell if they can be isolated. Okay. But the major differences in case of enzymes, if you increase temperature and pressure, what happens? If you increase temperature and pressure, so till now what we have understood, that enzymes work because of its shape, right? Because of its shape, certain substrate comes and binds to it. But if you increase the temperature and pressure, in that case, the shape of that enzyme disrupts. Okay, shape of enzymes disrupts. And if this shape will disrupt, what will happen? So enzyme will not function, right? So enzyme will not function. So it means that the increase in temperature and pressure is not good for the activity of an enzyme. But in the case of inorganic catalyst, what happens? Inorganic catalyst, if you increase temperature and pressure, usually the rate of reaction increases. Okay, rate of reaction increases in most of the cases. Okay, so this is the major difference between the enzyme and inorganic catalyst. The enzymes are very much sensitive to this temperature and pressure, and increase in this temperature and pressure will lead to the disruption of the function of enzymes. However, there are certain exceptions to this. So there are certain organisms or bacteria, for example, they lives in extreme conditions like high temperature, in high salt condition. So how does these bacteria survive there? So these bacteria or these organisms have suppose if it is surviving in high temperature, it means it has thermostable enzyme. This means that the enzyme can function at high temperature. So based on this adaptation, the enzymes have adapted themselves to function in such extreme condition. So there are certain exceptions. For example, some organisms have thermostable enzymes. Thermostable enzymes. Because this enzyme can function at, means this can function at high temperature and function at higher temperature. And the example is thermoacidophils. Thermoacidophiles. So that thermoacidophiles are what? These are bacteria that survive at higher temperature and they have this thermostable enzymes. So today for now, let's end it here. And in the next lecture, we will see about types of enzymes. And then we will also see graphical representation or how does an enzyme work, that we will see through equations and everything. Okay, that's all for now. Thank you.